Hi, IB Bio students. So today we're going to continue with 8.2 and we're going to be looking specifically at the electron transfer chain, which is part of aerobic cellular respiration, and we're going to look at the details of the electron transfer chain. Before we do so, I want to go and look at the overview of cellular respiration that we've learned before, and I want to focus on this summary here of ATP production. So if you look at this, remember that part of the ATP that gets generated in aerobic cellular respiration comes from substrate level phosphorylation. Well, that means that in the glycolysis process, there was a substrate or a substrate intermediate within this metabolic pathway here that was able to release the 4 ATP molecules. But remember, they translate into 2 net ATP molecule production because 2 ATP molecules were put in the process. So a substrate level phosphorylation happens here in glycolysis. Substrate level phosphorylation also happens in the Krebs cycle, where you get also 2 molecules of ATP. So if you add these up, you get 4 molecules of ATP made in the substrate level phosphorylation process of cellular respiration. But if you look at oxidative phosphorylation, which is what we're going to focus on right now with the electron transfer chain, keep in mind that all of these hydrogen electron carriers that we call taxis, they are going to pick up a hydrogen and electron, and they're going to take these hydrogens and electrons and take them to the electron transfer chain to generate a lot of ATP. So if you do the math here, you see that in this oxidative phosphorylation process that happens in the electron transfer chain, you're going to end up with 32 ATP molecules. So let's look at the math a little bit of these electron transports, or these taxis, or hydrogen carriers, as we can call them as well. If you are a carrier that is made in the cytoplasm of the cell, or in the cytosol of the cell, then you only have a capability to release 2 ATP molecules per hydrogen carrier. So if you do the math here, you see that 4 ATP molecules are released through these 2 hydrogen carriers that are released in the cytoplasm and that are then taken to the electron transfer chain. Now, if you are a hydrogen carrier that is made in the link reaction, for example here, then remember that the link reaction happens in the mitochondria. So any hydrogen carrier that is generated in the mitochondria has the ability to release more ATP molecules per hydrogen carrier. And in this case, it can release 3 ATP molecules per hydrogen carrier for a total of 6 ATP molecules that are going to be generated in the electron transfer chain. Same thing here, right? Since the Krebs cycle is in the mitochondria, this hydrogen carrier can release 3 ATP molecules per hydrogen carrier for a total of 18 ATPs released in the electron transfer chain. FADH2, remember, is not as common as NADH, and it can only generate 2 molecules of ATP per hydrogen carrier for a total of 4 ATP molecules generated in the electron transfer chain. So then if you do the math, 32 plus 4, that gives you a total of 36 ATP molecules per 1 glucose. So now let's focus on this oxidative phosphorylation process that happens in the inner membrane of the mitochondria. So let's look at the electron transfer chain. So the essential understanding kind of says a lot. The understanding says that transfer of electrons between carriers, remember carriers are the taxis, so electrons are being released from these carriers into the electron transfer chain in the membrane of the cristae, and then that is going to generate a proton driving force that is then going to be able to generate ATP. So a lot of things here going on in this understanding that we need to break up. So let's focus on the where right now. So where does the electron transfer chain happen? We know that it happens in the inner mitochondrial membrane, also known as the cristae, and we know that that membrane is folded to increase the surface area available for the transport chain. And we know that also protons that are going to get released through this process are going to accumulate in the intermembrane space, and that's what's going to generate a high proton force. So let's look at the steps here. Oxidative phosphorylation occurs in a number of distinct steps. So there are 3 steps that we are going to be looking at. So let's focus on the first step. The first step is to generate a proton motive force. Move this up a little bit. There we go. All right, so this proton motive force can also be called an electrochemical gradient. So let's first concentrate as to what we're looking here. Here we see the inner membrane. The inner membrane of what? The inner membrane of the mitochondria. So that means that somewhere here, there's got to be an outer membrane of the mitochondria, okay? And that means that this in the middle is the intermembrane space. And then that means that inside, we have the matrix of the mitochondria. So first we have to understand what we're looking at here, okay? So now that we know what's happening here, let's look at how these taxis or electron hydrogen carriers work. So we know from before that the glycolysis process released hydrogen carriers, so did the link reaction, and so did the Krebs cycle. So remember that NADH is not the only hydrogen carrier. FADH is also a hydrogen carrier, FADH2 actually, even though it's not being shown here. So what happens to these carriers? Well, look at them. They're getting oxidized. So oxidation is happening here. Remember that oxidation is to lose. So these hydrogen carriers are going to lose their electrons and their hydrogens. Now remember that these hydrogen carriers first had to get reduced, right? So NAD plus got reduced into NADH in the other processes of cellular respiration. But now NADH is getting oxidized. So again, oxidation is happening here. So hence the name oxidative phosphorylation, because these electron carriers are now losing their electrons and their hydrogens. So how do the electrons and the hydrogens play an important role here in this process? So let's look at it. So notice what's happening to the electrons. The electrons are going to bounce from one integral membrane or transmembrane protein to the next. So the electrons are going to go from one to the next, and to the next, and to the next, and to the next, and to the next. Hence the name, the electron transport chain. The chain is made up of integral transmembrane proteins. So when these electrons jump from one membrane that's embedded in the inner mitochondrial membrane, they lose energy, okay? So these are high energy electrons, right? But when they're transferred from one protein to the other, they lose energy. Now, that energy is not just going to dissipate. That energy is going to be put into good use. So these hydrogen ions that these taxis picked up and now released, these hydrogen ions, what are they going to do? They're going to get pumped with the energy released by the electrons. They're going to get pumped from the matrix of the mitochondria into the intermembrane space. And remember that before we talked about form fits function and we said that the intermembrane space was small, and we said that that was good. Yes, it's good because as these hydrogens get pumped from the matrix to the intermembrane space, they're able to generate a high hydrogen ion concentration. And this accumulation of hydrogen ions is what we call the electrochemical gradient or the proton motive force. Okay? So after these electrons have traveled and the hydrogen ions get pumped into the intermembrane space, we end up with an electrochemical gradient. What's going to happen? We've got to move on to step 2. So let's look at step 2 here. Step 2 is what we refer to as chemiosmosis. So chemiosmosis is the process where these protons are going to diffuse through ATP synthase, which is this green molecule here, and generate ATP. Okay? Now remember I told you that we were going to be able to generate 32 molecules of ATP, right? Because of all the electron carriers and the electrons that were released and the hydrogens that got pumped out. So let's look at this in more detail here. All right. So now you have a proton motive force here in the intermembrane space. But now these hydrogens diffuse back into the matrix of the mitochondria. So again, this is the matrix of the mitochondria. This is the inner mitochondrial membrane, or the cristae, and this is the intermembrane space. Intermembrane space. Okay? Here in the middle. So hydrogens are going to get pumped back in, and since these hydrogens, actually they don't get pumped back in, but they actually diffuse back in because you have a high concentration of hydrogen ions here and a low concentration of hydrogen ions here. They don't need to get pumped. So I used that word incorrectly, sorry. They will diffuse back in, going from high to low, okay? And when they diffuse back into the matrix, this is what we call chemiosmosis. And when these protons or hydrogen ions diffuse back in, right, they are able to diffuse back in because they go through this molecule called ATP synthase. ATP synthase. I can't spell. Synthase. Sorry. Synthase. All right. ATP, here it is. ATP synthase. So as the hydrogen ions diffuse through ATP synthase, right, they trigger the molecular rotation of the enzyme. So this molecule, this protein, this ATP synthase is an enzyme. And this molecular rotation of ATP synthase is going to generate the ATP. How much? 32 molecules of ATP per glucose, right? Now, we don't go into the mechanics of how this works. We just know that it works, and that's good for now. All right, so let's move on to step 3. And step 3 talks about the role of oxygen in all this process. So oxygen is needed to bind the free protons to maintain the hydrogen gradient, resulting in the formation of water. So let's look at how this is happening. So here we have step 3, which is reduction of oxygen. So in order for the electron transport chain to continue functioning, the de-energized electrons, so this is a de-energized electron, must be removed. So oxygen acts as the final electron acceptor. So again, remember that cellular respiration is an aerobic process, right? So oxygen is hanging out here in the matrix of the mitochondria. And we know that oxygen is very electronegative. It's the most electronegative, right? So it's able to call these electrons and attract these electrons. So oxygen is able to remove the de-energized electrons to prevent the chain from becoming blocked. This electron transport chain has to continue. It can't stop. So oxygen here, not only is it binding with the electrons, but it's also binding with the free protons in the matrix. So we have free protons in the matrix. Why? Because they were diffused back in, right? And remember that we always have to keep the high concentration of protons on the intermembrane space, and we have to keep a low concentration of hydrogens here in the matrix. So these hydrogens that are being diffused, they need to be removed as well. Otherwise, you're going to end up with a high concentration of hydrogen ions or proteins in the matrix, and it's going to disrupt the whole thing. So hydrogens also need to be removed. Electrons need to be removed. And the way to remove them is pretty simple, right? If you take two hydrogen ions, you couple them with two electrons, you get two hydrogen atoms. And if you take those two hydrogen atoms and you bond them with oxygen, you get water. So one way to remove the electrons and the hydrogens is to form water. And that's why water is a byproduct of the electron transfer chain. So here we see it, guys. C6H12O6 plus oxygen, 6 molecules of oxygen, makes 6 molecules of carbon dioxide. We saw the carbon dioxide being released in the Krebs cycle, plus 6 molecules of water. We see the water being released here in the electron transfer chain, plus 36 molecules of ATP. We see 32 ATP molecules here in the electron transfer chain and the other 4 in glycolysis and the Krebs cycle. So all in all, the cellular respiration equation, right, happens through these processes. The last process being the electron transfer chain. So make sure that you review this. Here's a good summary of oxidative phosphorylation. Reviewing the summary in distinct steps like this, it's very helpful because if there ever is a short answer question on a test to explain oxidative phosphorylation, then you have this as a concrete guide. And there is also a nice little video here that you should watch that shows you the overview of oxidative phosphorylation. It's also very helpful to watch these videos. And I'm actually going to play it right now because why not? Here it is. Electron transfer chain, ATP. Here come the hydrogen carriers. They release their electrons and their hydrogens. The electrons are going to travel down the electron transfer chain, lose energy along the way. That energy is going to be used to pump the hydrogens to the intermembrane space where there's high number of protons. They're going to diffuse back in through ATP synthase. That is going to allow ATP synthase to rotate and therefore phosphorylating ADP to ATP. And that's how you get the majority of ATP. Now, the hydrogen ions are going to, in conjunction with the electrons that need to be removed, form water. And there it is. All right, guys, hope you enjoyed this video. Go back and review your material, and that's it for today.